Okay, so today we are going to be going over the functions of all the things we labeled on the cell diagrams yesterday. Okay, so we're going to talk about what every single part of the cell does, okay, and kind of how that job happens. We're also going to look at how does the cell transport materials. Cellular transport is probably the most important concept in the biology unit of Science 10. Right, because it comes back not just when we're talking about single cells being able to transport stuff, but it's also the basis for how a multicellular organism transports materials, okay, over even larger distances. So okay, it's going to be applicable in the first half of the unit and in the second half of the unit when we're dealing with multicellular organisms. Okay, so we need to understand the significance of this compartmentalization. If you want to compartmentalize something, what are you doing to it? What are compartments? Storage places, right? Separate little rooms or areas. So if you compartmentalize something, are you breaking it into little rooms? Okay, that's what compartmentalization means. Okay, a cell is compartmentalized. That means each of those little organelles, okay, the structures we labeled yesterday, is its own separate compartment within the cell. And that means that in those compartments, we can create different environments. And that's important for those little organelles to do their job. Okay? Being compartmentalized is an advantage over having to do a bunch of jobs in the same area. Okay? So we're going to talk about a little bit more about compartmentalization in a little bit. Okay? We got to know the role of each organelle. That's going to come into play when you have your quiz tomorrow. Okay? Tomorrow you will get a diagram of the cell, and the questions will be, give me the name and function of the following numbers. Okay, like it could be like 1 to 12. Maybe I only ask you for half of them. Okay, but your job will be that you have to give me the name and function of the organelles of the cell. Okay, all right, third thing, understand that cells are a complex system of interdependent processes. Okay? Interdependent means that they depend on each other. Okay, and that if one link in the chain is broken, then the whole system will break down. Right, so we have to have everything working because step 3 depends on not just step 1, but it also depends on step 5, even though that may not make sense right now. It actually depends on things that come after it. Okay, so we have to have this interdependency of processes. Okay, number 4, understand how the cell carries out its functions. So we'll talk a little bit about kind of how each organelle does its job. And fifthly, understand how materials are transported across the cell membrane, okay, and within the cell. So like I said, this is probably the most important point, okay, in the entire unit. We'll be talking about essentially 3 main methods of cellular transport. And we have to understand how each one works, be able to identify it, explain what's going on. Questions so far? Okay. So, like we said, cells are divided up into compartments. Each organelle is its own compartment. Okay, so the mitochondria is a separate compartment, okay, from the Golgi apparatus, which is a separate compartment from the nucleus, and so on and so on. Okay, this is an advantage because the processes that go on in each organelle may require different conditions than the others do. For example, inside the lysosome is like inside your stomach. It's acidic and cellular material is being broken down chemically. Do you want that condition in the nucleus where your DNA is kept? No. Okay, so obviously then we should have separate rooms for those 2 jobs. Okay, it's just like in your house. The example here is, okay, like what you have in your house. You essentially have 3 separate temperature environments in your house. The environment in which you live, which you keep probably around 20, 21 degrees Celsius, because that's comfortable for you. That is not a good temperature to keep food from spoiling. So you have a fridge for that. Okay, you have your refrigerator. And in your refrigerator, the temperature is about 4 degrees Celsius. You wouldn't want the inside of your house to be 4 degrees Celsius because that'd be pretty darn cold all the time. Right? But it's a good temperature for keeping things fresh short term. Where do you put things you want to keep from spoiling long term? In the freezer, which is even colder. It's minus 17 or something like that. Okay, obviously you don't want to live with your house at minus 17. Okay, so you have a separate environment to keep to store food long term. Okay? Those three jobs can't be performed well in the same environment. If I was trying to do that, I would have to keep my house somewhere around like 8 degrees Celsius. Is it doing a good job of any of those three things then? Right? It's sort of doing all three, but it's not doing a very good job. You're cold because it's only 8 degrees in your house. Okay, food is still going to spoil faster than it would in your fridge, and you can't keep anything frozen at 8 degrees, so you've got no ability to store stuff long term. Okay, so essentially it doesn't work. Right? So being a cell like this is an advantage over being a cell without compartments. Okay? There are cells that don't have compartments. Okay? Cells that have compartments are called eukaryotic. Right? We are a eukaryotic organism because all of our cells have these compartments. Bacteria are prokaryotic cells. They don't have compartments. So they are like the house that's at 8 degrees Celsius. Okay? They are performing all the jobs that have to be done in the cell all in the same place. Okay? That means none of them get done very well. That's also why prokaryotes tend to be very, very small. And there are no multicellular prokaryotes. Okay? They can't do the jobs well enough to specialize, become big, or anything like that. Okay? That sort of makes sense, everybody, the importance of compartmentalizing. Okay? We have a separate environment for every job to be done, and it's being done in the perfect environment for that job. Okay. So know that word, eukaryotic. Okay, the other kind of cell, okay, is prokaryotic. Okay, those don't have the compartments. So maybe we'll just write prokaryotic, no compartments. Right, done with that one? All right, so to give you an idea here, remember I told you that prokaryotes are a lot smaller, okay? So this is a prokaryote, this is a bacteria, okay? This is an animal cell, a typical animal cell. You can't even see the whole thing. In fact, you're probably looking at like less than 1/10 of that cell, right? But you can see the whole bacterium easily, right? So prokaryotic cells don't get very large because they're not very efficient, okay? Now, on the same note, they're also not sensitive, not as sensitive as a eukaryotic cell, right? You can find bacteria in the most, let's say, inhospitable places. Like there are bacteria that live in the hot springs in Yellowstone National Park. Okay, like actually in the geysers that have the boiling hot water in them, right? There are some that live in the deepest part of the oceans, right? In the driest deserts, things like that. Like they can live pretty much anywhere because they're not quite so sensitive because they don't have to worry about maintaining these special perfect environments within the cell. They just don't do that, okay? Which actually makes them more able to adapt sometimes. Okay. All right, and then a virus obviously is very, very small, okay? But a virus, and this goes back to our definition of life, okay? If it's going to be alive, what did it have to be made up of? Cells. Viruses are not cells, okay? A virus is a small string of genetic material, either DNA or RNA, encased in a capsule made of protein. And that's it. A virus has no metabolism. It cannot reproduce on its own. Okay? Doesn't consume food, doesn't release waste, nothing. Okay? It is nothing more than an infectious particle. So there's a lot of debate as to whether a virus can be considered alive. By the standard definition of life, got to be made of cells, a virus is not a living thing. Right? Can a virus make you pretty sick? It can. How does it do that? It's not a living thing. How does it make you sick?
Does it kill off the normal cells?
The virus itself doesn't kill your cells. It's actually your own body that ends up killing the cells. Okay? A virus makes you sick by doing this. It would attach to your cell. Okay? So it would just come down here and attach to the membrane, and it would inject its genetic material into the cell. The only thing that's allowed to go into the nucleus is genetic material. So this genetic material that is the blueprint for making a virus goes into your nucleus. Okay? The nucleolus sees this thing come in and goes, oh, where does that go? And just kind of puts it in with the rest of your DNA. Okay? Shortly thereafter, the virus that's on the outside starts asking for something to be made, just like any other request from the cell. I need to make insulin, I need to make this, I need to make that. A request is sent saying, make this, right? And it's usually like a trick. It would be like it asks the cell to make, let's say insulin for an example. Okay? Instead of making insulin, the nucleolus sends out this new piece of DNA that it just got, which doesn't make insulin, it makes a virus. It's the blueprint for a virus. So your cell builds a virus. And then requests again to have the thing it asked for built. Okay? Because it made a virus and not what it asked for. So it makes another virus, and another one, and another one. Okay? And in the course of a few hours, that cell will fill up with viruses. Okay? Because all it's become is a virus factory. Okay? All it does is build viruses until it pops. Like it literally will pop because it's so full of viruses. And then each one of those viruses goes out and infects another cell. Okay? You ever had that experience where you go to bed and you got like the little tickle in your throat, and then when you wake up, you feel like you'd have to get better to die? Right? Like the virus can take you quick, okay? And literally within a few hours, because it can reproduce that fast because your body's doing all the work.
How do you kill viruses?
What your body does is it goes out and hunts down and kills all the infected cells.
Oh, with red blood cells?
Yeah. With like killer T cells, cytotoxic T cells, whatever. They all go out and they kill your own cells. That's why you feel so weak when you have a virus. Your body's fighting with itself. Okay? And it's a whole situation of, oh, you're infected. You have to die. Yeah. You have to take one for the team. Okay? And that's how your body deals with it. It's like, oh, I know that's one of our own cells, but it's gone bad. We have to kill it. Okay? And so you just get really, really weak because you have to destroy your own cells to get rid of the virus. Going out and hunting down the individual virus particles in your body is impossible. There's way too many of them. Okay? But once they're latched onto a cell, they're easier to get because they're stuck there. Okay? So they kill that whole cell. Any viruses that are inside of it are destroyed, and then they're gone. So it takes you a while to get over it. Okay? All right. That was an aside. It wasn't really. I'm never going to ask you how a virus works, incidentally. Okay. Within the nucleus, okay, we said the other day that the nucleus's job is just to hold your DNA. Okay, so inside the nucleus, DNA is stored. Right? Now, when the DNA is in your nucleus, it's really disorganized. It's just kind of strewn everywhere. How many people have ever seen like what chromosomes look like? You ever seen those? Sometimes you see like pictures of them, but okay, they look like fairly neat and organized. They only look like that when a cell's dividing. When the cell is actually functioning normally, it's like someone took a ball of yarn, completely unraveled it, and threw it in a pile. Okay, that's how the DNA looks when it's inside the nucleus and your cell is functioning normally, right? The nucleolus has to go and find within this snarl of DNA all the pieces it needs. It's amazing that anything ever gets done. Okay, but all of that DNA is stored inside of the nucleus, and that's the nucleus's only job: provide a safe place for the DNA to be. All right, so if we're looking at the nucleus under an electron microscope, okay, under like not much magnification, it just looks like a ball. Okay, kind of almost like a ball that's maybe got pimples on it. Okay, if you zoom in a little bit more, those little pimples are actually pores. Okay, they're actually little holes that can open and close to allow the copies of the DNA that the nucleolus makes to escape the nucleus. That's how they get out. Right? Nothing else can get in. Just those copies of DNA can get out. Right? And then the surface of the membrane is actually an interlocking kind of series of fats and things like that and fibrous material that keep it strong. Okay, so job of the nucleus, if I was writing it on a quiz and I wanted to have just a short, dirty definition, it would be the nucleus, 2 words. Stores DNA, good enough. Okay, that's what the nucleus does. If you're writing it on a quiz, that's all you need to write. Nucleus stores DNA. Okay. All right. DNA is used to make materials. You only have to understand this in kind of a basic entry level in Science 10. DNA is a code, okay? And that code can be read by certain parts of your cell. If your cell needs something, that section of the code is copied and sent to the ribosomes. The ribosomes read the code and make the material. That's really the extent to which you need to know it. Okay? So information is copied, sent out of the nucleus, and then used to make materials. Right? The thing that's important about that is that is the nucleolus's job. The nucleolus copies the DNA. Okay? And in as much as it copies the DNA, it's also then kind of in charge of protein synthesis. Okay? The ribosomes actually make the protein, but the nucleus is kind of in control of it because it's the one that sends out the instructions. Okay, so you can see on the diagram here, right, this blue double-stranded thing is a piece of your DNA. If a certain material needs to be made, the nucleolus will send out this stuff called RNA polymerase, which is just a chemical essentially that can pull your DNA apart so it can see one of the two strands. Okay? It'll copy one side of that, and that's what it's doing here. Okay? So you can see that it's been pulled apart, and now the blue is being copied into pink. Okay? So this pink is a single-stranded copy of your DNA because the DNA is complementary. Right? One side it determines the other, right? A always goes with T and C always goes with G, right? You guys learned that in Science 9, right? Okay. So it makes a copy of it and then puts it back together. The copy then goes out through one of those little pores that we saw on the last slide, okay, and goes to a ribosome. The ribosome can read the code and put together the protein. Then it's done. Okay. That piece of DNA is then either sent back to the nucleus or it's just broken down, okay, into its raw materials again. Questions on that? So what does the nucleolus do? That's what we get off the slide. What does the nucleolus do? Copies DNA. Good enough. Okay. Nucleolus copies DNA. Okay. A lot of the interior volume of the cell is actually endoplasmic reticulum, either rough or smooth, but there's a lot of it in there. Okay. And the reason for that is it has a pretty important job, or actually many important jobs. Right? It's often drawn like this, it's just kind of a squiggly material. The rough ER is obviously covered with the little ribosomes, which is what makes it appear rough, but also a bit more organized. The smooth ER doesn't have any ribosomes on it, and it's just kind of a big snarl. Under an electron microscope, this is actually what it looks like. The smooth ER would be kind of this side, okay, and you can see that it's pretty random looking. But the rough ER is a lot more organized, and you can see all the little ribosomes stuck on its surface, whereas over here you see very few, if any, ribosomes. Okay, everyone kind of follow the difference? Okay, so in the smooth ER, okay, so if we're looking at what does the smooth ER do, okay, the smooth ER makes lipids, okay, and the lipids are used for repairing membranes, okay. Complex carbohydrates are broken down. Right, so starches, if you had like a bunch of rice or a bunch of pasta or something like that, that's really starchy food, or potatoes, really starchy food. Okay, those starches get broken down in the smooth ER, and then the simpler sugars can be used. Okay, drugs and poisons are detoxified. So the smooth ER's got a lot of jobs. Okay, it breaks down complex carbohydrates. It makes lipids. Okay, it breaks down poisons and detoxifies toxins, and makes hormones. So it has a lot of jobs. So it's a very, very important organelle. If your cell was to lose it, it would die. Not that it would die if it lost any other organelle either, but okay, obviously there's a lot of jobs being done, okay, by the smooth ER. Okay. The reason we want to do these jobs in the smooth ER is because the smooth ER is often connected directly to the cell membrane, which means if we've broken down drugs, poisons, or other toxic materials, they can go straight to the membrane without touching any other part of the cell, which is good because they're toxic and poisonous, and we wouldn't want them to interact with any other part of the cell. Okay. The complex carbohydrates, when they get broken down, because the smooth ER is kind of everywhere, they can quickly get to like the mitochondria where they'd be needed to be broken down and used in energy. Okay. Lipids, we want those to get to the membrane quickly because they're used to repair the membrane, so we want them to get there fast. Okay. So that's kind of the logical part of all this stuff happening in the smooth ER. Okay, hormones, lots of times cells are producing those not for themselves, but for other cells. So we need those to get to the membrane quickly so they get out of the cell and where they need to go. All right, so basically any job that involves getting something out of the cell quickly has to be done there. Okay, questions on the smooth ER? Okay. All right, so we said here, lipids have to be moved quickly to the needed sites, poisons and drugs detoxified and quickly removed. Okay, travel through the ER limits contact with the rest of the cell. All right, now I don't think we said anything about the rough ER here, so we need to say what the rough ER does. Ooh, green's bad. Okay, let's try black. Okay, rough ER transports protein. Okay, that's all the rough ER does. That's why it's covered in ribosomes. The ribosomes make the protein, the rough ER transports it where it needs to go. Okay, that's also why it tends to be really close or surrounding the nucleus. That means that the copies of the DNA that have the instructions don't have to go very far. Okay, so the cell's organization is a lot more kind of thought out than it might appear when you look at one. Okay, questions on the two types of ER? All right, Golgi apparatus. We said yesterday the Golgi apparatus is like the garbage man of the cell. Obviously, that's not the answer we want to write down on a quiz or a test. Okay, what we would like to say is that they excrete waste products. Okay, that's mostly their job. Okay, so some of the products of the ER go to the Golgi for excretion. Okay, but essentially it makes vesicles, which are little like membrane enclosed sacs that can go straight to the membrane and get out. Right. So remember I said yesterday, it's making these vesicles. It's kind of like how you carry your garbage out of the house, right? You don't carry your garbage in your hands with no bag, okay? Because it spills everywhere. You put it in a bag and you carry it outside. Well, this is the thing that puts the garbage in the bag, okay? And then sends it out to the garbage can without making a mess, right? So let's say for the Golgi apparatus, if we're looking for a quick kind of function, we would say excretes wastes. Now, weird thing about the Golgi is that it has two specific sides. Waste products and other things that it needs to package come in on one side and get exported from the other. Okay, so it has a receiving side and a shipping side. Okay, so the cis face, you'll never need to know that, what it's called. Okay, is the receiving side, so stuff comes in here, it gets processed inside the Golgi, and it leaves in these little vesicles. Okay, these little bubbles that then go to the membrane, and they essentially, when they get to the membrane, they just pop. Okay, and they become part of the membrane, and whatever was in them is outside the cell now. Okay, everybody all right with that? Yeah. All right, we talked about the lysosomes yesterday. We also talked about the peroxisomes yesterday. So the lysosome, okay, has really strong membranes because it has to protect everything from the digestive chemicals within it, right? So the job of the lysosomes basically is to digest food. And we're only going to find those in animal cells, right? Chemoheterotrophs, more accurately. Okay, but they'll only be in organisms that actually have to consume organic material for food. Okay, plants aren't going to need these. All right, now lysosomes don't just digest food; they also help to recycle damaged materials in the cell. So if you had an organelle, like a mitochondria or something that was malfunctioning, not working anymore, the cell is the ultimate recycler. It'll just make a lysosome, send it over, and eat it. Okay, and by eating it, essentially, it's breaking it down into its raw materials to use the raw materials again. Since the whole thing isn't working, okay, we're kind of just picking away the parts that can still be used. Okay, questions on the job of the lysosome? Right. Then we have the mitochondria. So the mitochondria is where cellular respiration occurs. Okay, so we might want to write cellular respiration. Okay, that's kind of the complex way of saying it burns sugar for energy. Because the reactions that go on inside the mitochondria yield energy, they also give off a lot of heat. So this is an organelle that needs two membranes. And it needs two membranes because it's got to be kind of like a thermos. It has to insulate itself against the rest of the cell. If it doesn't, the heat that it will generate could damage other parts of the cell or cause them to not function properly. Okay? As an example, if you have a fever, how do you feel besides hot and cold? Do you feel energized or tired? Yeah, you just feel bagged, okay? Because your body is running way faster than it's supposed to, and it's driving your temperature up. Okay? Most of your bodily processes, especially the small cellular level ones, work best at 37 degrees Celsius. When you start running a fever, those processes don't work as well. And as a result, your body doesn't as efficiently make use of its energy. So you feel really, really tired. Okay? And if you run too high a fever for too long, it can kill you because those simple processes that are keep you alive won't work. Okay? They won't work outside of that kind of narrow temperature range where we feel comfortable. The same thing happens if you have hypothermia. Okay? That is, if you get too cold and your body temperature starts to drop, these same processes don't work very well. Okay? And as a result, you get tired. Okay? That's usually what kills people that die from freezing to death. Okay? They start to feel really, really tired. And they just sit down, fall asleep, and freeze. Okay? Happens on Mount Everest and K2 all the time. Okay? People have to be like, no, you got to keep going. You got to keep walking. Don't sit down, because if they sit down, they'll die. Okay? Because they just get so tired. Same thing happens when you have a fever. Our body works only in a narrow range of temperatures. So at a cellular level, we don't want the extra heat that this thing can produce to get out. Okay? If it does, it'll interfere with the proper functioning of the cell. So it has a double membrane to insulate the cell against it. Okay, all right, questions on that? Okay, and again, guys, every one of these pictures is showing kind of like the artist's representation, and then this is what it looks like under a microscope. Okay, are we going to see most of these when we look through the microscope? No. Okay, what we'd be able to see under the microscope would be like the nucleus, because it's pretty big. You can see chloroplasts. Sometimes you can see the rough ER. It often looks like it's right next to the nucleus. It kind of looks like the nucleus has a beard. Okay, so if the nucleus is a face, it'll have like kind of a little triangle kind of coming down from the bottom of it. It'll look like the nucleus has a beard. That's the rough ER. Okay, if you're really lucky in a plant cell, you can see the water vacuole, okay, things like that. But for the most part, much of this stuff is too small for us to see. Okay, speaking of the plant cell, okay, the chloroplasts are again one of the bigger organelles, so we can see those. Okay, and when we look at the plant cell on Monday, okay, I'll kind of explain to you what they look like and what to look for. But they're going to be they're generally in a real cell, kind of greenish in color, okay, and they contain what looks like under an electron microscope, that's this here, like kind of stacks of coins almost, or checkers or whatever, okay, so they're stacked up like that, and these are called thylakoids, and this is where the process of photosynthesis really takes place. Okay, the chloroplast also has a double membrane, because the process of photosynthesis, okay, can also produce some heat. These are obviously the things absorbing light, and light's a form of energy. So the more light you absorb, the hotter you're going to get. Okay, so that has to be insulated against the rest of the cell as well. Okay, so if I was looking at kind of a quick definition or function of the chloroplast, it would be this. Carries out photosynthesis. Like really any definition of what the chloroplast does should include the word photosynthesis. That's really what it does. Okay, other organelles within the cell. Okay, kind of just quick ones here. Okay, the cell membrane, oddly enough, is probably the most important organelle. Without it, there'd be no cell. Okay, but the cell membrane isn't this like layer of cellophane that we kind of envision it to be, like this kind of seals up the cell. It's actually quite a bit more complex than that. Okay, it looks like this. Okay, so there's lots of fats. That's what all these kind of little blue and white spheres with the tails on them are. Okay, those are all fat molecules, but it's more like a Jell-O salad. Okay, embedded within the matrix of the cell membrane, which would be like the Jell-O, are like pieces of fruit. If we're taking that Jell-O salad analogy that far. Okay, those pieces of fruit are things like globular proteins, okay, different types of proteins, receptor molecules. Your cell membrane is covered with what are effectively antennas, okay, that can receive chemical signals from your body that tell the cell it needs to do certain things. Okay, most of those chemical signals are hormones. Your cells respond to hormones in certain ways. If your cell receives makes contact with an insulin molecule, the cell membrane becomes more permeable to sugar and helps to absorb the sugar. Okay, if we're looking at if adrenaline comes into contact with the cell, okay, that signals the cell to ramp up its metabolism and start doing things faster, okay, to give us more energy for our fight or flight response, things like that. So there's basically a receptor or an antenna for every possible chemical signal the cell could receive. Okay? Unlike the antenna in your car, which can receive a whole bunch of radio stations, the cell membrane has an antenna for every station, which is weird, but that's kind of the way it works. Okay, because they're not a radio signal, they're a chemical signal, and they actually fit together based on their shapes. So there's an antenna with a shape for this molecule, an antenna with a shape for that molecule. Okay, so it is quite a bit more complex than we typically envision it being. Okay? There's a whole bunch of stuff embedded in the membrane. All right, so what does the membrane do? If I was looking for a definition of the cell membrane, it would need to contain these two words. Okay, controls endocytosis and exocytosis. Endocytosis means movement into the cell. Exocytosis means movement out of the cell. Right? That's what the cell membrane does. Okay. The last thing I want to see as a definition of what the cell membrane does is protects the cell. Okay. I don't want to see that. Okay. That's not technical enough. I want to see those two words, controls endocytosis and exocytosis. Okay. Movement into and out of the cell, either across or through the membrane. Okay. The cell wall, going to be present only in plant cells. Okay, now, if I was telling you yesterday, if you recall, okay, the cell wall is more like a frame, okay, and this is actually what it looks like. Right, so here's the cell membrane, this purple stuff, and it's solid, okay, but the cell wall is hollow. And if we looked at it from the other side, if we were able to turn it this way, it would look exactly the same as this looking on it that way. It's just like a framed house, okay, that actually hasn't had the walls per se put on yet or had the insulation put in or anything like that. You could walk right through the walls of the house at that stage. That's what the cell wall looks like. So it's just a frame that the water vacuole can push against to help support the cell. Right? So if I was writing a definition of the function of the cell wall, okay, it would need to include something about being a frame, okay, that supports or keeps the shape. Right? And then, as we just said, the water vacuole kind of works in concert with the cell wall. Okay? It's going to push from inside against that frame to keep the cell rigid. Right? We saw kind of what happens when the plants don't have enough water, they wilt, and then when we give them water, the water vacuoles fill back up and they regain their original shape. Okay? So if I was writing down a definition for water vacuole, I would want to include those two words, turgor pressure and supporting the cell. Okay, we talked yesterday as well about the centrioles, okay, and that they are only in animal cells and that they help with cell division. Okay, and this is how they do that. Put the marker down. All right, so when a cell is getting ready to divide, okay, it's going to send the centrioles to either end of the cell. And the centrioles will then put out these little fibers. They almost look like cables, okay? And the cables will join up with the chromosomes, okay? That is the genetic material that all lines up right in the middle of the cell before a cell divides, okay? Those cables will then pull the chromosomes apart, ensuring that each half of the cell gets a copy of it. Okay, because the first thing a cell does before it splits is copies all the DNA. Okay, so then it pulls them apart and makes sure that each side gets what it needs. Everyone follow me there? And we said the reason that an animal cell has to do that and a plant cell doesn't is an animal cell can change shape. Okay, whereas a plant cell is much more rigid and nothing can really interfere with it while it's dividing. Whereas for an animal cell, if it changed shape suddenly during cell division, the DNA might not get to the right place otherwise. Okay. All right, and then microfibers and microfilaments, okay, that's the cytoskeleton, right? So we might just want to write cytoskeleton beside that. Okay, and they just help the cell stay intact during changes in shape. Okay, again, they're basically only part of an animal cell. All right, so we're going to look now at the different types of transport within the cell. Okay, there are what we call the big three. Okay, the big three are diffusion, osmosis, and active transport. Those are the three we're going to go over in the most detail. Okay, those are not the only three ways that stuff gets transported, but they're the big three. Okay, so we'll be focusing mostly on those. Those are the ones that you'd be asked about on an exam or a quiz. Okay, more than kind of the minor ones. All right, so we're going to talk about diffusion first because diffusion is by far the most common way that a cell moves material within the cell. All right, in fact, everything naturally moves by diffusion. Okay? Everything has a natural tendency to go from a place where it is very concentrated to a place where it is less concentrated. Everything moves away from being concentrated to less concentrated. Okay? We call it, it works on the fart principle. Okay? If you are right next to a person when they fart, the experience is quite acute. Okay? The smell is very strong and very obvious. Okay? But if you are farther away from that person, the experience is, well, less unpleasant. Because by the time the fart molecules have diffused through the air and gotten to you, they are far less concentrated. And so you get like a hint of it, but it's not terrible. Okay? I know it seems gross, but you won't forget it. Okay? It kind of works on that. All right? So diffusion is this natural movement of things away from an area of concentration. You can kind of see that in this picture here, where they're just injecting dye into water, right? And it naturally moves away from the point where it's being injected. Okay? And it's becoming, as you can see, less and less concentrated the further away from the injector site it is. right? So cells naturally use this. And the reason they naturally use it is because it's free. It costs no energy at all from the cell to move materials by diffusion. And as long as we're talking about movement within the cell, diffusion is pretty effective. It works very well over very small distances. It does not work well over large distances, because the further something has to diffuse, the less concentrated it becomes. Okay, it's kind of like the shotgun approach. Okay? It's just going to spread out, and there's hardly going to be anything if it had to travel any distance at all. But since we're talking about a cell, which is very small, diffusion works really well over those kinds of distances. Okay, are there some drawbacks to diffusion? Yes. If it has to go really far, it doesn't really work very well because it takes too long and it would not be very concentrated when it got there. Okay, so those are kind of the two big drawbacks. The big benefits: free, works great over small distances. Right? That's why your cell uses diffusion, okay, to move things within the cell. All right, so simple rule of diffusion, okay, in the absence of other forces, something moves from where it is more concentrated to where it is less concentrated. We call that down the gradient, okay, because that's how naturally things go, is downward. It doesn't require any energy to go downward. Okay, so diffusion is passive. Okay, it requires no energy from the cell. Okay, so let's say that within this cell, this organelle right here is producing some material. Okay, over here in this organelle, okay, is where that material is needed. Okay, so here is where that material will be very high in concentration. Over here, since it's already been used up, is where it would be very low in concentration. So is the material naturally just going to move from this one to this one? What do you think? By diffusion, will it kind of naturally move in that direction? It will, but it'll also naturally move in any direction where it's not concentrated. So while, yes, probably most of it, so like, let's say here, the material is really, really concentrated, okay? By the time this process is done, sure, some of it gets here, but some of it ends up over here, and some of it ends up over here, okay? It's not perfect. It's not targeted. It's not like, you know, if you FedEx something to somebody, okay, it goes from one place directly to the other. It doesn't work that way. Okay? It's more like Canada Post, where, I'm sorry, I shouldn't say that, okay? Where sometimes it gets there and sometimes not. It's not addressed to where it needs to go, okay? But it'll generally get there, generally. Okay? All right, questions on how diffusion works? All right, so like we said, this is fine as long as these two things are essentially together in a small cell. If this cell gets really big, this process won't work, right? It'll try, but the distances over which it has to go are too big. It won't be very concentrated when it gets there, and it'll take a really long time. Okay, so it's not a fast process, it's not a targeted process, but it's free and works good over small distances. All right, so again, this is just within the cell. Okay? Diffusion works within the cell. It's not transporting across the membrane, just it's already in the cell or it's produced in the cell or whatever, it's going to move by diffusion. Okay. Second process of the big three is osmosis. Okay? Probably a word you've heard before, but more probably along the lines of reverse osmosis. How many people have heard that term before? Okay, if you buy like certain types of bottled water, Dasani, Aquafina, okay, ones like that, those are RO. Okay, they've been filtered through the process of reverse osmosis. Okay, osmosis, the natural process, is the process by which water, and only water, moves through the membrane or across the membrane. Okay, so one thing we have to remember is that osmosis is only going to transport one material, water. Okay, it's not going to transport anything else, just water. Okay, and it is also passive. Okay, that means it requires no energy. The cell doesn't have to invest anything in this process. It's just going to happen. That's good and bad, the fact that it's just going to happen. because it also means the cell can't control it. Okay? It's just something that happens. So most of the time, what osmosis does is maintains a good balance of water on both sides of the membrane. Okay? Water naturally wants to move from an area where there's very little salt to an area where there's a lot of salt. Okay? It naturally wants to move in that direction. So if there's a lot of salt, let's say in my bloodstream, water is going to move out of my cells and into my bloodstream. Okay? That's going to mean that my cells kind of become dehydrated. All right? And eventually, if I get too much water in my bloodstream, my kidneys are going to start filtering it out, and I'm going to lose a lot of water. This is why you're not supposed to drink salt water. Okay? Like if you're stranded in the middle of the ocean, don't drink the water for two reasons. Osmosis will kill you, and secondly, the ocean is the world's toilet. If you've seen Finding Nemo, all pipes go to the ocean. Don't drink the ocean water. Okay? Yeah. Okay, so we don't want to drink ocean water because it's full of salt. If I drink that salt water, the salt will end up in my bloodstream, in my cells, whatever, and it's going to start pulling water out of my tissue. Okay? And then I'm going to start peeing more to get rid of all the salt. Okay? The water is being pulled out to get rid of the salt, and then my kidneys are getting rid of the salt along with water. So for every liter of salt water I drink, I'm going to lose 1.5 liters of my own water. Okay? So it's actually going to make things worse. If you drink salt water, you're going to get thirstier, okay, than if you hadn't drank any. So you're better off to not drink anything. Okay? In fact, for the first day or two, you're better off to drink your own urine than to drink the ocean water. It'll actually have less salt in it. I know that seems really gross, but okay, you'd be better off doing that than drinking the ocean water. Okay, so don't drink salt water. It will dehydrate you really, really quickly. Everyone kind of follow on that? Yeah, okay, and it's osmosis that'll do that. So essentially what we have going on is this. If I have, and this is something you might do in Bio 20, okay, if I have a kind of a U-shaped test tube, and then across the middle, I have essentially a membrane of some kind. What we usually use when we do this experiment is sausage casing. Okay, sausage casing is actually made out of intestine. So it's made out of cells. Works just like a cell membrane. Okay, on one side, I put fairly concentrated salt water. On the other side, I put like very weakly or normal like tap water. Okay, what'll start to happen is water will move across the membrane from this side to this side. The salt will stay. The salt doesn't go. Okay, just the water. And the water goes over to this side, and you'll see the volume go down on one and up on the other until the concentration of salt is equal on both sides. Once the concentrations have been equalized, then there's this state of equilibrium and osmosis stops. Everyone kind of follow me on that? Okay. What happens if you put salt on a slug?
Doesn't it like die?
Yes, but what happens to it as it dies? Anyone seen it? It shrivels up. Okay? Because a slug doesn't have the same kind of epidermal or skin layer that we do. If you put salt on it, osmosis just starts to happen really rapidly. Okay? It's got salt on the outside of it, and water just starts coming out of it to balance the salt concentration. The slug can do nothing to stop it. Okay? And so it just sucks the moisture out of the slug, and the slug just shrivels up and dies. Don't do that. It's cruel. Okay? So your friend harms animals?
No, she's like 7. She
Still
just like
kind of scary. Yeah. Okay. That's kind of where, yeah, a little bit scary. Okay. All right. So everyone kind of followed. That's osmosis. That's kind of what happens with that. It would depend how long it had been going on for. Maybe a couple minutes, yes. But you remember, you can't just throw a slug in the water; it'll drown too. It's got to be able to breathe. So, could you rinse the salt off and maybe it would live? Yes, something like that. But no, just dumping it in a cup of water is going to kill it too. Okay. You guys are really starting to freak me out a little. Okay, so yeah, that's essentially what happens. If we look at that from a cellular level, okay, this is what goes on with osmosis. In normal conditions where the salt inside and outside the cell are equal amounts of water move in and out. Okay, water is always moving by osmosis. It doesn't like really stop, but the net movement stops. Okay, if I put it in concentrated salt solution, water gets moving right out of the cell because there's more salt outside of the cell than inside, and the cell just shrivels up like a slug would. Okay, the opposite is also true. If I was to put a cell in just plain distilled water, the cell would burst because there's salts inside the cell and there's no salt in distilled water. So osmosis is going to just start pulling water into the cell because there's salt inside and there's no hope of balancing it because there's no salt outside. So water just keeps rushing into the cell and the cell just keeps getting bigger and bigger till it pops. Okay? The cell can't stop this process. Okay? It'll just go until there's a balance or the cell is destroyed, one or the other. Okay? All right, so so the things that we need to remember about osmosis: it's passive, it's only water, okay, and it's across the membrane, okay, to balance salts. Okay, it's trying to balance salt concentrations. Okay, most of the time it's not a problem. The process just kind of goes merrily on, right? But it is one of the triggers for our thirst reflex. Okay, this is kind of a trick that like restaurants and bars will play on people. Okay, you guys haven't been to the bar because you're not old enough, but okay, typically like the food you can buy at a pub or something like that is very salty. Okay, and they want you to eat the salty food because it makes you thirsty. Okay? It causes this osmotic imbalance that causes water to move out of our tissues and into our bloodstream. Your body detects that. Brain detects the change in water in the bloodstream and signals the thirst reflex. Hey, we're losing water. You got to have something to drink. Okay? It's a nifty trick, but okay. Movie theaters, same thing. Salty snacks. Okay, salty snacks make you buy more pop. Okay, buy more water, whatever. Okay, makes you thirsty. You buy more stuff. Okay, it's one of those things that people take advantage of. Okay, questions on osmosis? I'm pretty good on how that works. Okay. So that's number 2 of the big 3. Okay, diffusion's number 1 of the big 3. Okay, osmosis number 2 of the big 3. Active transport is number 3 of the big 3. It is the exact opposite of the other 2. Okay, the other 2 processes were both passive. They required no energy. The cell really couldn't stop them, speed them up. It has no control over them. Okay, active transport is the active pumping of solutes against the gradient. So it goes the other way. Okay, where the other 2 processes are seeking balance, active transport produces imbalance. Okay, it is pumping things the other way, moving water uphill, whatever. Okay, it is pumping against the gradient. In order to do that, it has to use energy. Okay, the cell expends energy in this process, and it is pumping the materials across the membrane. Okay, now when we say pumping, we're only talking about very small, like single molecule scale. Like mostly it's electrolytes, calcium, sodium, potassium, okay, things like that that your muscles and nerve cells need. Okay, but mostly that's what they're moving. They're not moving like sugars and proteins and really big molecules. It's just pumping small stuff. Okay, so we were saying that the cell membrane is like this Jell-O salad, okay, kind of thing. Well, some of the proteins that are embedded in the cell membrane are what we call carrier proteins or transport proteins, and they can change shape when energy is applied to them. They essentially act like gates, okay, and they are part of the active transport process. When the cell needs to transport something actively, it expends the energy to keep changing the shape of these proteins, and they eventually start acting like a pump as they change shape. Okay, so we can see here that this is outside the cell, this is inside the cell here. Right, so we have the open position of the carrier protein. Ions will come in there. Okay, energy will be expended, and the carrier proteins reverse themselves. They change shape to close off and effectively pump the materials in, and they'll keep doing that until they've pumped in as much material as is needed. All right, so active transport, really good in that it can get stuff done quickly, it can go against the gradient, but its drawback is that it requires energy. It's not passive, it's active. Okay. All right, so it's a major factor in the ability of the cell to maintain concentrations of small molecules. Okay, questions about active transport. All right, so these three processes, like we said, they're kind of natural processes. The cell uses them very often. They don't involve the membrane having to make really large changes in shape. Okay, obviously in active transport, the little carrier proteins change shape, but the membrane itself stays kind of the same. Okay, what we're going to talk about now are some kind of minor things that the minor transport methods, that is, that involve actually major changes in the shape of the membrane in order for them to happen. It's how the cell takes in big stuff, right? The cell doesn't need to take in big stuff very often, okay? So these processes are, like we said, quite a bit less commonplace than these ones are, right? Because the cell is transporting small stuff, lots, okay? Big stuff, less often. Okay, exocytosis means to move stuff out of the cell. Okay, if something is going out of the cell, it's either coming from the Golgi, most often, or the smooth ER. In either case, it's going to arrive at the membrane in a vesicle. Okay, that little membrane enclosed sac. Okay, when it merges with the membrane, okay, it effectively becomes part of the membrane, and whatever was inside is expelled. Okay, expelled out of the cell. If that's waste product, then it's expelled out of the cell. The blood will pick it up, and the blood will take it to the liver or the kidneys or wherever it needs to go to be gotten rid of. Okay, if it's a hormone, okay, then we call that secretion. So it's coming from the Golgi apparatus. Same process. It's in a vesicle. The vesicle merges with the membrane, pops the stuff out into the space between the cells. The blood picks it up and takes it wherever it needs to go. Okay, that's exocytosis, movement out of the cell from the Golgi or the smooth ER. Okay, but it involves these vesicles. Okay, so exocytosis, okay, secretes large molecules by fusion of the vesicles with the membrane. Okay, remember, exocytosis not as important as the big three. Okay, so that's how the cell can get large molecules out of the cell. Sometimes the cell needs to get large things into the cell, right? So there's three ways that it can do that. Okay, endocytosis is what we call, in general, movement into the cell by changes in the membrane. Okay, endocytosis into the cell. Remember we said the cell membrane's function was to control endocytosis and exocytosis. So these are the processes by which those happen. Okay, so there's three processes. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. They all involve the membrane changing shape to engulf something. All right. So how many people have a dog? Okay, you ever have to go pick up after your dog? You take him for a walk, right? And you got to put the thing over your hand and pick up the poop. Okay, that's how phagocytosis works. Okay, you have the membrane over your hand, you engulf the stuff, you pick it up and you turn the bag inside out. Now it's inside a vesicle. Okay, that's what's going on here. Okay, except it's food, right? So the cell will engulf the material, take it inside in a vesicle. Okay, basically turns the membrane inside out, and that material goes into the cell and can be transported to a lysosome where it can be digested. Right? But it can't just, a piece of big food like that can't just work its way through the membrane. It has to be engulfed. It's too big to pass through. Okay. Pinocytosis, same process, but it's for taking in large amounts of water very quickly. Okay? If osmosis can move stuff fairly quickly, but if a cell is really dehydrated, it's going to gulp. Okay? So osmosis would allow a cell to like slowly sip water, this is chug-a-lug. Okay, like I'm so thirsty, I'm going to drink a big like gallon of water right now. Okay, kind of thing. So it's you're saying this is like chugging. This would be the osmosis would be just sipping slowly. All right, but it's exactly the same process. The membrane changes shape. Fluid goes into the little kind of vesicle. The vesicle closes off, goes into the cell, and quickly we've got water inside. Okay. Receptor-mediated endocytosis, again, mechanically the same. The membrane is closing in and engulfing something, but this time it's a chemical messenger. So we said that in the case of hormones, okay, there's an antenna for basically every hormone. So if that signal is received, then the antenna signals the cell membrane to engulf the hormone because hormones are too big to go through. Okay, so they get engulfed and then they go into the cell where the membrane or the vesicle can be punctured, and then the hormone can go to whatever part of the cell it needs to go and affect. Okay, so that's how hormones come into, because they're big protein molecules that can't be absorbed. Okay. So like we were saying here, for phagocytosis, we engulf a food or solid particle. Okay? This is how your white blood cells kill bacteria. They eat them. Okay? They go along, they find the bacteria, they just engulf them, and they digest them with a lysosome, and they're dead. Okay? Only really your white blood cells can do that, other cells can't. Okay? For pinocytosis, like we said, it's just going to be changing the shape of the membrane to engulf fluid. Okay? This is what it actually looks like under an electron microscope. You can see that the membrane has kind of bent inwards and there's fluid trapped inside, and you can see the different stages of it. This one here is ready to kind of pinch closed. Okay, this one here looks like it's kind of just opening. Okay, and they're taking in fluid. Okay. And then we have the receptor-mediated endocytosis. Okay, and so we've got the little receptors. When they receive their signal, the cell membrane kind of pushes in and then pinches closed, and the vesicle goes in. And you can see that really well in these electron microscope pictures here. So this, if you look really closely, okay, you can actually see here's a receptor molecule and here's the hormone sitting on it. Okay, so I've drawn that on there. I'm just going to erase what I've drawn so you can actually see it in the picture. Okay, so there's a whole bunch of those along this part here, and once it you can see here the membrane is about to close off. There's just a very narrow gap here. You can see all these receptors are full. They all have a molecule, okay, these kind of circular looking things, okay, sitting on them that are being taken in. All right. Okay, what are the big three again? Osmosis, diffusion, and active transport. Okay, these three that I just went over, these are minor. Okay, I will barely ever ask about these. Okay, if you're getting a written response question on a test, it's about the big three. Okay, it would be about either diffusion, osmosis, or active transport. I'm never going to have a large written response question about endocytosis. Okay. All right. I want you guys to answer those 4 questions. Complete sentences, please. Okay. Some of them, the answers are directly in your notes somewhere, and some of them you'll have to think about it a little bit. Okay. But I want you guys to answer those, and then we'll go through them here in a minute or two. Okay. Let's have a look at these. So, if you're colorblind, it's because your cells can't produce a certain protein. And then that makes the receptors in your eye not work properly, and you can't see certain colors. So where in your cell is the problem? That's usually what we want to say, because we think automatically the ribosomes make protein, so the problem's with the ribosomes. Where do the ribosomes get their information from? From the nucleus. So the problem is actually in your DNA, which is why color blindness is genetic. Okay? If you are colorblind, okay, the part of your DNA that tells the cells and the ribosomes how to make this certain protein is wrong. They actually make a protein, but it doesn't work. Okay, same thing with being diabetic. You have a section of your DNA that codes for insulin, but it's wrong, and your cells make this other protein that doesn't work. Okay, so if you're colorblind, it's genetic. Okay? And in fact, men are far more likely to be colorblind than women because the gene for colorblindness is on the X chromosome, and men only have one of those, whereas women have two, so they are far more likely to have the correct copy of that gene because they have two chances of it, whereas guys only have one. Okay? Mr. Van Erve, just down the hall, he's colorblind. Like, totally colorblind. Like the other day, he and I were wearing a shirt that had the same pattern. He's like, hey, we're wearing the same shirt. I'm like, sure we are. They were totally different colors. He had no idea. Okay? No idea. And you can tell if he gets out of the house before his wife sees him because he's wearing stuff that doesn't match. You got poor guy who just can't see color at all. Okay? And that's why actually we have things set up in our society to kind of prevent that from being a problem. You ever notice how red lights are always on the top or on the left? Green lights are always on the bottom or on the right because some people can't tell the difference between red and green. Okay, but by the position, they know whether the light is red or green. Okay, things like that. Okay, all right. Why is it important for organelles to have their own membranes? It's a big long word, started with C. Oh, we better look that one up. It was in the key points, like the first key point. Evan. Compartmentalization. It's important because every organelle has a different environment in which the process it does works best. Okay, with every organelle having a compartment, those processes work more efficiently. Okay, if the lysosomes membrane ruptured, what would happen to the cell? It would eat itself. Yeah. Okay. We'll see you guys tomorrow.